old when dies, at different rates and different tissues. Growth, development, maintaining health, and healing from diseases require an intricate interplay between the rates of coordination of cell division and death. Mitosis and cytokinesis, which divide the DNA and the rest of the cell respectively, and apoptosis is a form of cell death. Cell cycle. Cell cycle is a series of events describe the sequence as a cell prepares for and undergoes cell division. Many division enable a fertilized egg to develop into a many trillion cell organism, and the picture in the presentation is one of the example of mitosis and apoptosis mold a body. So as you can see, cell number increase and decrease from apoptosis, in which in the embryo, apoptosis normally carves the fingers and toes from the web structure. So as you can see in the picture in the presentation, apoptosis fails to carve the digits and weaving persists as is this hand. Cell cycle has two major stages, which is the interphase and the mitosis. In interphase, the cell is not dividing, and in the mitosis, cell is dividing. The first stage of cell cycle is interphase. During the interphase, a cell continues the basic biochemical functions of life while also replicating its DNA and some organelles. Interphase is divided into two gaps, which is the G1 and the G2 phases, and one synthesis, the S phase. So the diagram in the presentation, during the G1 phase, a cell resumes the synthesizing of proteins, lipids, and carbohydrates. The synthesizing of macromolecules and biomolecules during the S phase helps to contribute the extra plasma membrane that required to surround the two new formed cells from the original one. During the G1 phase, the period of the cell cycle varies the most in duration among the different cell types. The dividing of cells during the G1 phase speed through 16 to 24 hours, and cells in the early embryo may skip G1 entirely. During the S phase, the cell replicates its entire genome. After the DNA replication, each chromosome consists of two copies of genomes that join in an area called the centromere. So as you can see in the presentation, the blue chromosome and the center area of that is called centromere itself. In human cell, S phase takes 8 to 10 hours. Many proteins also synthesize in this phase, including those the mitotic spindle that during the phase will pull the chromosome apart. During the phase of G2, it occurs after the DNA has been replicated but before the mitosis begins. During this phase, more proteins are synthesized in order to a membrane to assemble made during the G1 phase are stored as small, empty vesicles beneath the plasma membrane. This vesicle will emerge with the plasma membrane that will ensure the boundary that will enclose with the two daughter cells. So as you can see in the picture in the presentation, chromosomes are replicated during the S phase. The two genetically identical chromatids of replicated chromosome joined at the center of the centromere. The cell cycle is continual, and the other stage of cell cycle is mitosis. In mitosis, cell distributes its content into two daughter cells with a complete set of chromosomes. Mitosis maintains the set of 23 pairs of characteristic of human somatic cells. Mitosis has 4 stages, which is prophase, metaphase, anaphase, telophase, plus the cytokinesis. The first stage of mitosis is prophase. As you can see in the picture, DNA coils tightly. This coiling shortens and tightens the chromosome, easing their separation. Microtubules assemble from tubulin building blocks in the cytoplasm, forming the spindles. So as you can see in the picture of prophase, at the end of prophase, the nuclear membrane breaks down, and the nucleolus is no longer visible. The next stage of mitosis is metaphase. As you can see in the picture, chromosome attach at the spindle fibers at the centromeres along at the center of the cell, which is called the equator. The next stage is anaphase. So as you can see in the picture, the sister chromatid separates, the centromeres is also divides, and the sister chromatids moves to the opposite poles itself. In telophase, the final stage of mitosis. As you can see in the picture, cell looks like a dumbbell with a set of chromosome at each end of it. The stage of telophase, the spindle fibers disappear, and the nucleoli and the membrane around the nuclei reforms at each end of elongated of cell, and the division of genetic material is now complete. The next is cytokinesis. Organelles and macromolecules distributed between the two daughter cells. Control of the cell cycle is the group of interacting proteins function in specific times in the cell cycle. They ensure that all the chromosomes are correctly replicated and apportioned into daughter cells. The cell cycle has 3 checkpoints, which is the DNA damage checkpoint, the apoptosis checkpoint, and the spindle assembly checkpoint, in which these 3 checkpoints is ensuring that the mitotic events occur in the correct sequence. First, the DNA damage checkpoints. It temporarily pauses the cell cycle while the special proteins repair the damaged DNA. During the apoptosis checkpoint, proteins called survive and override, telling the cell to die in order to mitosis division will occur rather than the apoptosis death cell. Later, during the mitosis stage, the spindle assembly checkpoint, ensuring that the construction of spindle and chromosome to it is perfectly checked and correct. So how can a cell know how many divisions remain? The answer lies in a chromosome called telomeres. The function of telomeres is like a cellular fuses that break down pieces are lost from the ends. Also, hormones control the cell cycle, in which the growth factors signal the cell from the outside, and the interaction of 2 types of proteins, the cyclin and the kinases, also triggers the mitosis inside of the cell. In apoptosis, a receptor in the plasma membrane receives a death signal, in which the apoptosis begins and the enzyme called caspases activates and tear apart the cell in an orderly fashion. Apoptosis is the program of cell death. If a cell undergoes apoptosis, it helps for preventing and eliminating the cancer spreading of the cell and also maintaining the tissue of homeostasis, in which eliminating the unwanted cells.